Hello, my name is Joshua Mugabe. I welcome you to Cell Structure. What is cell theory? The cell theory asserts that the cell is the constituent unit of a living being. Before the discovery of the cell, it was not recognized that living beings were made up of building blocks like cells. The cell theory is one of the basic theories of biology. Are there living beings without cells? Viruses are considered the only living beings that do not have cells. Viruses are constituted by genetic material, that is DNA or RNA, enwrapped by a protein capsule. They do not have membranes, cell organelles, nor do they have self-metabolism. In 1665, Robert Hooke, an English scientist, published the book Micrographia, in which he described a piece of cork viewed under the microscope presented small cavity similar to pores, which were filled with air. Based on later knowledge of what were the walls of those cavities constituted, what is the historical importance of that observation? The walls of the cavities observed by Hooke were walls of that plant cells that form the tissue. The observation led to the discovery of the cells, a fact only possible after the invention of the microscope. In that work, Hooke established the term cell, now widely used in biology, to designate the cavities seen under the microscope. What are the two big groups into which cells are classified? Cells are classified as eukaryotic or prokaryotic. Prokaryotic cell is that without a delimited nucleus. Eukaryote cells are those with the nucleus delimited by membrane. Do bacteria cells have a nucleus? In bacteria, the genetic material is dispersed in the cytosol, and there is no internal membrane that delimits a nucleus. Are there any bacteria made up of more than one cell? There are no pluricellular bacteria. All bacteria are unicellular prokaryotic. What is plasma membrane of the cell? What are its main functions? Plasma membrane is the outer membrane of the cell. It delimits the cell itself, a cell interior with specific conditions for the cellular function. Since it is selectively permeable, the plasma membrane has an important role for the passage of substances inwards or outwards. What are the chemical substances that compose the plasma membrane? The main constituents of the plasma membrane are phospholipids, proteins, and carbohydrates. The phospholipids, amphipathic molecules, are regularly organized in the membrane according to their polarity. Two layers of phospholipids form the lipid bilayer, with the polar part of the phospholipid pointing to the exterior of the layer, and the nonpolar phospholipid chain in the interior. Proteins can be found embedded in the lipid bilayer, and there are also some carbohydrates bound to proteins and to phospholipids in the outer face of the membrane. What is the difference between the plasma membrane and cell wall? The plasma membrane and cell wall are not the same thing. Plasma membrane are called cell membrane. Is the outer membrane common to all living cells. It is also made of phospholipid bilayer, embedded proteins, and some appended carbohydrates. Because the cell membranes are fragile, in some type of cells, there are even outer structures that support and protect the membrane, like the cellulose wall that is in plant cells, and the chitin wall in some of the fungi cells. Most bacteria also present an outer cell wall made of peptidoglycans and other organic substances. What are the main respective constituents of cell walls in bacteria, protists, fungi, and plants? In bacteria, the cell wall is made of peptidoglycans. Among the protist algae, the cell wall is made of cellulose. In fungi, the cell wall is made of chitin, the same substance that makes the exoskeleton of arthropods. In plants, the cell wall is made of cellulose. Do membranes form only the outer wrapping of the cells? Lipid membranes do not form only the outer cover of cells. Cell organelles such as Golgi complex, mitochondria, chloroplasts, lysosomes, endoplasmic reticulum, the nucleus are delimited by membranes too. What type of cell came first in evolution? The eukaryotic cell or prokaryotic cell? This is an interesting problem of the biological evolution. The most accepted hypothesis asserts that the most simple cell, the prokaryotic cell, appeared earlier in the evolution than the more complex eukaryotic cell. The endosymbiotic hypothesis, for example, affirms that aerobic eukaryotic cells appeared from the mutualistic ecological interaction between aerobic prokaryotes and the primitive anaerobic eukaryotes. Concerning the presence of the nucleus, what is the difference between animal and bacterial cells? Animal cells, cells of living beings in kingdom Animalia, have interior membrane that delimits the cell nucleus, and thus they are eukaryotic cells. In these cells, the genetic material is located within the nucleus. Bacterial cells, cells of living being in the kingdom Monella, do not have organized cellular nuclei, so they are prokaryotic cells, and their genetic material is found dispersed in the cytosol. What are the three main parts of eukaryotic cell? The eukaryotic cell can be divided into two main portions: the cell membrane that separates the intracellular space from the outer space, physically delimiting the cell. The cytoplasm, the interior portion filled with the cytosol, the aqueous fluid inside the cell, and the nucleus. the membrane delimited internal region that contains the genetic material. What are the main structures within the cell nucleus? Within the cell nucleus, the main structures are the nucleolus, an optical dense region, spherical-shaped, where there are concentrated ribosome, RNA, associated with proteins. There may be more than one nucleolus in the nucleus. The chromatin, made of DNA molecules dispersed in the nuclear matrix during cell interphase. The karyoteca, or the nuclear membrane, the membrane that delimits the nucleus. What are the substances that constitute the chromatin? What is the difference between chromatin and chromosome? The chromatin dispersed in the nucleus is a set of filamentous DNA molecules associated to the nuclear proteins called histones. Each DNA filament is a double helix of DNA and thus a chromosome. What is the fluid that fills the nucleus? The aqueous fluid that fills the nuclear region is called karyolymph or nucleoplasm. In the fluid, there are proteins, enzymes, and other important substances for the nuclear metabolism. Of what substances is the nucleolus made? Is a membrane around the nucleolus? The nucleolus is a region within the nucleus made of ribosomal RNA and proteins. It is not delimited by membrane. What is the name of the membrane that delimits the nucleus? To which component of the cell structure is that membrane contiguous? The nuclear membrane is called karyotheca. The nuclear membrane is continuous with the endoplasmic reticulum membrane. What are the main cytoplasmic structures present in the animal cell? The main cytoplasmic structures of the cell are centrioles, the cytoskeleton, lysosomes, mitochondria, peroxisomes, Golgi apparatus, endoplasmic reticulum, and ribosomes. What are cytoplasmic inclusions? Cytoplasmic inclusions are cytoplasmic molecules, aggregates, such as pigments, organic polymers, crystals. They are not considered cell organelles. Fat droplets and glycogen granules are examples of cytoplasmic inclusion. Where in the cell can ribosomes be found? What is the main biological function of the ribosomes? Ribosomes can be found free in the cytoplasm, adhered to the outer side of the nuclear membrane, or associated to the endoplasmic reticulum membrane, defining the rough endoplasmic reticulum. Ribosomes are the structures where protein synthesis takes place. What is the difference between smooth and rough endoplasmic reticulum? The endoplasmic reticulum is a delicate membranous structure contiguous to the nuclear membrane and present in the cytoplasm. It forms an extensive net of channels throughout the cell, and it is classified into rough and smooth types. The rough endoplasmic reticulum is a great number of ribosomes attached to the exterior side of its membrane. Smooth endoplasmic reticulum does not have ribosomes attached to its membrane. The main function of the rough endoplasmic reticulum are synthesis and storage of proteins made in the ribosomes. The smooth endoplasmic reticulum plays a role in the lipid synthesis and in muscular cells. It is important in the conduction of the contraction stimulus. A net-like membranous complex of superposed flat circulates of vesicles detaching from the extremities seen in electron microscopy. What is the observed structure? What is its biological function? What is being observed is the Golgi complex or the Golgi apparatus. This cytoplasmic organelle is associated with chemical processing and modification of proteins made by the cell, and with the storage and branding of these proteins for posterior use and secretion. Vesicles seen under the electron microscope contain material ready processed, ready to be exported or secreted by the cell. The vesicles detach from the Golgi apparatus, travel across the cytoplasm and fuse with the plasma membrane, then secreting their substances to the exterior. On which organelle of the cell structure does the intracellular digestion depend? What is the chemical content of these organelles? Intracellular digestion occurs by the action of lysosomes. Lysosomes have digestive enzymes, hydrolases, that are made in the rough endoplasmic reticulum and stored in the Golgi apparatus. Lysosomes are hydrolase containing vesicles that detach from the Golgi apparatus. Why are lysosomes known as the cleaners of the cell wastes? Lysosomes carry out autophagic, heterophagic digestion. Autophagic digestion by digesting residue substances from cellular metabolism. Heterophagic digestion by digesting substances that enter into the cell. Lysosome unfold the substance to be degraded by forming digestive vacuoles or residue vacuoles that later migrate toward the plasma membrane, fusing with it and liberating or exocytosis the digested material to the exterior. Which are the cell organelles that participate in cell division and in the formation of cilia and flagella of the eukaryotic cells? The organelles that participate in cell division and in the formation of cilia and flagella of some eukaryotic cells are the centrioles. Some cells have cilia, like paramecium, the bronchociliated epithelium, and many others. Or flagella, like flagellate protists, sperm cells, and many others. These cell structures are composed of microtubules originating from the centrioles. Centrioles also make the aster microtubes that are very important for cell division. What are the morphological, chemical, and function similarities and differences between lysosomes and peroxisomes? Similarities. Lysosomes and peroxisomes are small membranous vesicles that contain enzymes and enclose residue substances from internal or external region, degrading them. Differences. Lysosomes have digestive enzymes, hydrolylase, that break substances to be digested into smaller molecules. Peroxisomes contain enzymes that degrade mainly long-chain fatty acids and amino acids, and that inactivate toxic agents including ethanol. Within the peroxisomes, there is the enzyme catalase, responsible for the oxidation of the organic compound by hydrogen peroxide. And when this substance is in excess, by the degradation of the peroxide into water and molecular oxygen. What are mitochondria? What is the basic morphology of these organelles, and in which cells can they be found? Mitochondria are organelles in which most important part of cellular respiration occurs, that is ATP production. Mitochondria are organelles delimited by two lipid membranes. Their inner membranes invaginates to the interior of the organelle, forming cristae that delimit the internal space known as mitochondria, and where the mitochondria DNA and mitochondria RNA, mitochondrial ribosomes, and respiratory enzymes can be found. Mitochondria are numerous in eukaryotic cells, and they are even abundant in these cells that use more energy, like the muscle cells, because they have their own DNA, RNA, and ribosomes. Mitochondria can self-replicate. Can mitochondria be considered the power plants of aerobic cells? Mitochondria are the power plants of aerobic cells because within them the final stages of cellular respiration processes occur. Cellular respiration is the process of using organic molecule, mainly glucose and oxygen, to produce carbon dioxide and energy. The energy is stored in form of ATP, adenosine triphosphate molecule, and later used in other cellular metabolic reactions. The mitochondria, the two last steps of the cellular respiration, take place: the Krebs cycle and the respiratory chain. What is endosymbiotic hypothesis about the origin of mitochondria? What are the molecular facts that support the hypothesis? To which other cellular organelles can the hypothesis also be applied? It is presumed that mitochondria were primitive aerobic prokaryotes that were engulfed in mutualism by the primitive anaerobic eukaryotes, receiving protection from these beings and offering energy to them. This hypothesis is called the endosymbiotic hypothesis on the origin of the mitochondria. The hypothesis is strengthened by some molecular evidence, such as that the fact that mitochondria have their own independent DNA, protein synthesis machinery, in their own RNA and ribosomes, and that they can self-replicate. The endosymbiotic theory can be applied to chloroplasts too. It is supposed that these organelles were primitive photosynthetic prokaryotes because they have their own DNA, RNA, and ribosomes, and they can self-replicate too. What are the main components of the cytoskeleton? The cytoskeleton is a network of small tubules and filaments distributed throughout the cytoplasm of eukaryotic cells. It is made up of microtubules, microfilaments, and intermediate filaments. Microtubules are formed by molecules of protein called tubulin. Microfilaments are made of actin, the same protein that participates in the contraction of the muscle cells. Intermediate filaments are made from protein too. What are the functions of the cytoskeleton? As the name indicates, cytoskeleton is responsible for the support and the normal function of the cell. It also acts as a facilitator for substances, transport across the cell, and for movement of cellular organelles. For example, sliding between actin-containing filament and the protein myosin. create pseudopods. The cells of the phagocytic defense system, like macrophages, cytoskeleton, is responsible for the plasma membrane projections that engulf the external material to be internalized and attacked by the cell. What are chloroplasts? What are the main functions of the chloroplasts? Chloroplasts, these are organelles present in the cytoplasm of plants and algae cells. Like mitochondria, chloroplasts have two boundary membranes and many internal membranes and sacs. Within the organelle, there are DNA and RNA and ribosomes and also pigment chlorophyll, responsible for the absorption of the photic energy that is used in photosynthesis. The main function of the chloroplast is photosynthesis, the production of highly energetic organic molecule, which is glucose, from carbon dioxide, water, and light. What is the molecule responsible for the absorption of photonic energy for photosynthesis? Where is the molecule located in the photosynthetic cell? The chlorophyll molecule is responsible for the absorption of the light energy for photosynthesis. These molecules are found in the internal membranes of chloroplasts. What are the colors of the electromagnetic spectrum absorbed by plants? What would happen to photosynthesis if the green light waves that reach the vegetable were blocked? Chlorophyll absorbs all other colors of the electromagnetic spectrum, but it practically does not absorb green. The green color is refracted, and such refraction provides the characteristic color of the plants. If the green light that reaches the plant is blocked and the exposure of the plant to other colors is maintained, there would be no harm to the photosynthesis process. Apparent paradox. Green light is not important for photosynthesis. There is a difference between optimum color frequency for the two main types of chlorophyll, chlorophyll A and chlorophyll B. Chlorophyll A has the absorption peak at approximately 410 nanometer wavelength annual, and chlorophyll B has the absorption in 450 nanometer wavelength blue. Which path is followed by the energy absorbed by the plants to be used in photosynthesis? The energy source of photosynthesis is the sun, the unique central star of our planetary system. In photosynthesis, the solar energy is transformed into chemical energy. The energy of the chemical bonds of the produced glucose molecule and of the released molecular oxygen. The energy of glucose is then stored as starch, a glucose polymer. It is used in cellular respiration process and transformed to ATP molecule. ATP is consumed in metabolic processes that spend energy. For example, in active transport across the membranes. Of what substance is the plant cell wall made? Of which monomer is it made? The plant cell wall is made of cellulose. Cellulose is a polymer and whose monomer is glucose. There are other polymers of glucose, like glycogen and starch. What is the function of the plant cell wall? The plant cell wall has structure and protective functions. It plays an important role in the constraint of the cell size, preventing the cell to break when it absorbs a lot of water. What are plant cell virtues? What are their functions? What is the covering membrane of the virtues called? Plant cell virtues are cell structures delimited by membranes, within which there is aqueous solution made of various substances like carbohydrates, proteins. In young plants, many small vacuoles can be seen. Within the adult cells, most part of the internal area of the cell is occupied by a central vacuole. The main function of the vacuoles is the osmotic balance of the intracellular space. They act as an external space inside the cell. Vacuoles, they absorb and release water in response to cellular metabolic necessities by increasing or lowering the concentration of the osmotic particles dissolved in the cytosol. Vacuole also serve as storage place for some substances. The membrane that delimits the vacuole is called tonoplast, named after the osmotic function of the structure. Thank you very much for participating in cell structures. Please do like, share, and subscribe.